The pseudopods are the long, slender extensions from the big ball-shaped cell in the center. And then, Sarah, finish it. What does it form at the end of one of its pseudopods?
A loop.
It'll eventually loop like this. But take a look at one of the unrolled ones, because it forms, each pseudopod at its end forms a pseudopod flap. Take a look at that big obvious one right down towards 6 o'clock on the page. Now that flap then does what, Sarah, to make it Schwann cell-like?
It wraps around the axon.
It wraps around the axon many, many times, forming, Sarah, a loop, a cover that we would call then a myelin sheath. So one oligodendroglial cell goes to 20 to 40 different axons that are going by it, and in that immediate area, check it out from the picture here, forms an outer loop or wrapper called the myelin sheath. What doesn't it possess? Labeled nowhere here. What doesn't it have? Somebody? Loud enough? No neural lemma. No outer thin membrane from the oligodendroglial cell, so no powers of nerve repair. This would be, for instance, looking at a portion of the brain. As we focus into the brain, we can find the oligodendroglial cell kind of nestled in the axons there, and it takes care of just that immediate area. Look down here. You have another oligodendroglial cell down at the bottom at about 7 o'clock, and its pseudopods and pseudopod flaps take care of that region. And there'd be another one out here, and another one down here, and another one up here, and so on. So that just takes care of the neurons in its region. Questions on that? Same type idea. If you cut one of these in half, cross-sectional view, it'd look like this. It would have that multi-layered look that you found in the white handout sheet before, picture C. Only it wouldn't be a Schwann cell, the oligodendroglial cell. How many nuclei would you have for 20 to 40 pseudopods? One. There's only one cell. How many nuclei do you have for 20 to 40 Schwann cells? 20 to 40, whatever the number is. Just in that alone, you've saved space. Just in the saving of the space of the nuclei. And I said the oligodendroglial cell was there to be more compact, take up less space. So you'd save it just on the nuclei, let alone the reduction in the number of mitochondria and Golgi bodies and everything else that would have to be in this cell to keep it alive. Questions on example 5? Anyone? Okay, those 5 cells then together are known as the neuroglia cells. The only other neuroglia structures that we would have inside the brain, just list them here as number 6. Put other neuroglia tissue. And I'm only going to mention it. By now you should understand it. They would be blood vessels, capillaries, and connective tissue that helps hold everything in place. We've talked all about the circulatory system up there, so we don't have to go into that, circle of Willis and all that business. But it supports, and the connective tissue would be things like fibrous connective tissue, adipose tissue for protection. All the different types of connective tissue would be up there serving as neuroglia tissue. And I don't want to make a big deal of it other than mention to make sure you know it's there, and it's got to be there in order for the systems to work correctly. Part B. When we started this, we said we're going to just divide Roman numeral 2 into 2 parts. A was going to be quick and simple, 5, 6 examples here of neuroglia tissue. Part B is called the neurons, or the neuron. And it's going to be lots of information on the cellular structure of the nerve cell. So that we get this squared away right at the beginning, why don't you put, even before you put number 1, that a neuron and a nerve cell are the same thing. Neuron and a nerve cell, same thing. Listen to what's different. A neuron and a nerve are different. A nerve is thousands of neurons collected together in a bundle. A nerve cell is the same thing as a neuron. So if a doctor tells you you've severed a nerve, that's serious, because you've cut thousands and thousands of pathways of information either coming from or going to your brain. If he says you've cut a nerve cell, not serious. One individual neuron isn't going to make or break the nervous system. And that could happen if you had a needle jabbed in your arm to give you a shot or a vaccine. That's not going to ruin the nervous system. A nerve could do a lot of damage. You could be paralyzed, for instance, if it was a whole nerve that was severed. Number 1, under Part B. And Brian, you're watching the clock for me today because we won't hear the bell. It's going to be 11 after. So give me a timeout signal at about 10 after. Okay, please. Number 1 is going to be structure of a neuron. And you want to take the first paper we had, the salmon-colored paper here, and take a look at that, please. And very quickly, we're going to go through the main parts of a neuron. And then we'll take a look at some of its fine details. On that paper, you should find some things lettered for you, A, B, and C. And as I've mentioned before, but you didn't really have to write it down for any good reason, now I'd like you to, and I've left you some space along the side of the paper there. You can use a big arrow pointing down towards the bottom of the paper. Mine is different color, but just so that we're talking about the same thing. An arrow pointing this way with the head of the arrow pointing down. Okay, and you can put the 3 parts that I'm going to go over along that arrow. A, the dendrites, is where an impulse enters a nerve cell, way out here on the ends. You can put that at the top of the arrow. B, the second part of a nerve cell or neuron, is called the cell body. You can put that kind of like in the middle of the arrow. And C, and look how long this is. Many, 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 many times longer than the rest of the cell. The axon is the third part. You can put that down at the end of the arrow where the head of the arrow is, and you've just given yourself the direction that an impulse travels through a nerve cell, always. From dendrite to cell body to axon. That little chart you just made on the side of your page gives you the direction of travel of an impulse, always. It never goes the other direction. You can't send an impulse axon to cell body to dendrite. It won't work. It can only go in the direction that I've just shown you. So always, you line up the neurons. So if you want to send a signal towards the brain, that would mean a sensory impulse. Where would the dendrites be? Near the brain or near your skin? Near your skin. The cell body would be just inside of it, and the axon let's say it's coming from my fingertip then, would come the length of my finger, my hand, my arm, my shoulder, my chest cavity, and into my spinal cord. That means that some axons, which are going to be the longest part, would be as long as from my spinal cord, into my spinal cord, down across the gluteal, through the femoral, down behind the ham, down the calf, around the heel, and out to my longest toe. That would be about the length of the longest axons. So axons can be tremendously long. Measure them in feet. I mean, that distance there has got to be 3.5 feet. On Manute Bol, that's got to be 5 feet long. Guy's 7'6" tall. So the taller you are, the longer your longest axons are. Dendrites tend to be short all the time. Now, if you were sending a motor signal from your brain out to your muscles, where would the dendrites be? Remember the direction of travel now. Where would the dendrites be? Out at your toe muscle or back touching your brain? Or which way you send the impulse? Yes, Sarah. Yeah, the dendrites would be plugged into the brain, the cell body would be there, and then you'd have this long axon traveling from the brain, let's say, out to the end of your fingertip or down to the bottom of your toe. That makes sense? The direction of travel always has to be in the way I've showed it. Katie?
I thought the nerve impulse went from like cell to cell. Is that there's only 2 cells involved?
Okay, good question. I mentioned before that impulses go from cell to cell, right hand being neuron A and left hand being neuron B, and there was a little space. That's within the central nervous system. Once a nerve fiber leaves the central nervous system and goes into the PNS, except for in a couple cases, it's always on a single neuron.
From like 2 cells are only involved?
Right. That's right. All this synapse to synapse business is inside the brain and the spinal cord. For me to send a message from the top of my brain down to the bottom of my spinal cord, I may have to go through 50 different neurons and 50 different synapses. When I get to the bottom of my spinal cord and then finish sending the signal to my toe, 1 neuron. So CNS, lots of neurons, lots of synapses. PNS, few nerves, and 1 synapse. And there's only one exception to that, and that is in the ANS. In the ANS, in order to send a signal out, it involves 2 neurons instead of 1. So when a message is coming out, it goes to the first neuron, then a little relay station, then to the second neuron, and to the organ that it's going to. Okay? Good question. Lots of neurons, lots of synapses, CNS. 1 nerve in most cases. The exception, ANS. 2 nerves in the PNS. So that's why I'm saying they have to be very long cells. Take a look at that picture, and you'll find that way down at the very end of the axon, get down near the bottom of the page, you can find these things called the terminal fibers. The terminal fibers are branches that come off of the axon. What are they trying to do? What is the structure of the neuron trying to do to the signal when you get right to the end of the axon? Do what to the signal? Spread it out. Hit a bigger surface area. Your axon doesn't end like this. Here comes the axon down my arm, and my finger is part of it, and there's one point that it sends the message. Oh, we got this neat ability. Right at the end, the axon flares out. So we haven't sent 15 axons down. We've sent 1. But right at the end, it spreads out into terminal fibers, and at the end of each terminal fiber, take a look at your handout sheet, there's something called a bouton. I think I've always wanted this. A person from France in my audience to tell me what the word bouton means. It's French, right?